So what will you do after you have done your wet lab? So there are three main steps in the NGS bioinformatics pipeline. The first one is primary analysis. So what is the primary analysis? Depending upon your sequence specific formats, you will get a raw sequence file. For example, in Illumina sequencer, you got what is known as a BCF file, BCF file, and that will be converted to a FASTQ file. And then in the secondary analysis, as was discussed by Francis in his talk, you are going to align these sequences to a reference genome. So if you're looking at a variant in the for a rare disease, you will align your reads or the sequence reads to the reference genome and identify variants. But when you're doing an exome, typically you get lots of variants. So then the final part is the tertiary analysis, where from a annotated VCF file you will filter down your variants to a handful of variants which you can manually check, which will fit the phenotype of the clinical phenotype of your patient. So this is what is secondary analysis. So the primary one was from the raw to the sequence file. So what we mean by alignment is that you are aligning it to your reference genome, but you should be very careful while aligning that the build should be the same as on which your exome or your panel was built. Otherwise, you might get a mismatch. So typically, it could be your HG38 or HG19, and then when you align the first you get a SAM, that is a sequence alignment map, and then you get a binary alignment, which is a compressed form of a SAM, and finally you get a variant calling file unannotated, which has to be annotated. So,
also, Amitji, just one more thing. Since you are in PGI, so you always get only human sample. But also remember like this organism aligning. I have personally aligned human data to mouse or mouse data to human, and then it was just don't think, just think like it is a.
Yeah, so it is important that you also choose the genome of like what organism you are working on. And in the tertiary analysis, as I said, that you will have lots of variants. So you will filter out according to different strategies. The first one is probably how common is that particular variant in the normal healthy population. So the more common a variant in the population, the less likely that it is going to be a pathogenic or a likely pathogenic variant. So, but the problem is that you don't have population databases for all populations. So mainly the databases where you have got database of genomic variation in healthy individuals, supposedly healthy because they are not actually healthy. They were having like chronic lifestyle diseases like hypertension or diabetes, and these were sequenced, and their genome or exome is available. So you see whether those variants that you have found in a patient are already present in these databases. But the problem is that these are there's underrepresentation of certain ethnic races or ethnicities in this population. So this is the tertiary analysis for a germline. You have to ascribe that variant to the particular phenotype, or you can like classify it as likely benign, uncertain, likely pathogenic, or pathogenic. I'll just focus on the benign and the germline variants because that's what we do. But in a somatic variants are the variants which you're doing in cancer specimens. So example, if you have a cancer specimen, you will do somatic variants, and there the classification is slightly different. There is a classification, a tier based system is there, which is different from what you do for a germline variant. So these are some of the steps that you can do. If you have some gene or a class of genes, you could write a code that I want to look at only these genes. For example, if you know that this is a particular phenotype like intellectual disability, neurodevelopment, or it is an inborn error of immunity, you could look only at those handful genes. Or for example, if it is a antibody deficiency or something like that, you could only select those genes and look at that. You could look at the population frequency, as I said, or the location. You can specify that you can only you want only to look at the coding regions. I mean, because those are the ones which would be more relevant. And then the phenotype. So how does the phenotype match with that particular variant? So the phenotype is very important because in a high throughput sequencing method, you get a lot of variants, but then you have to see whether the variant that you have found on your analysis, whether that it fits with the clinical phenotype of the patient. And then the mode of inheritance. So, example, if it is a autosomal recessive disease, you should find two variants. It should be homozygous, so you have to have one on each allele, and if it is autosomal dominant, then obviously a heterozygous variant. And for X-linked, you have to get X-linked variants in X-linked genes. So, what are the resources and tools which are available? This is variant effector predictor, NOR, Exomizer. These tools can be used to, annotate your VCF files. And then the genome build is very important, as I've said, like where you are going to align your sequence reads. Then the transcripts are also important, because for each gene you can have multiple transcripts, and some labs might use, so the labs in Europe, they use the Ensemble transcript, whereas in other places the RefSeq or the NMIDs are used for when you are referring to a transcript of a gene. And there are standards for nomenclature. So, for naming a variation, for classifying a variation, it is HGVS, and transcript, there is something known as main. So, this was a project between NCBI and Ensemble, because there was a discrepancy between the transcripts. So, matched annotation, NCBI and Ensemble. So, this is the main transcript, which is very important. This was because the same transcript would be referred by a different name in NCBI and by a different name by Ensemble. So, this was then, and then the gene name also, you've got standardized nomenclature. So, this is very important, like the build is very important, the transcript you are referring to, and in transcript some labs will mention the Ensemble transcript, some will mention the NCBI transcript. So, both of these are very important. And sometimes visualizing the variant on a IGV or integrative genomic viewers becomes very important, especially in complex variants like indels. So, we have seen that sometimes when there is a complex indel, the software will call it as two variants, whereas when you look at the BAM file on IGV, you can clearly see that so viewing the variants, especially the complex variants on IGV, is very important. And it can be it is free to use. So, there are different versions of IGV you can download on your either Mac or Windows, and then look at the files there. So, this is wherever there is a variant, it is colored, the nucleotide is changed. For example, here you see at this point, the A is the reference one and it is changed to T, and but it is not in all the reads actually. So, a heterozygous typically would be 50%, and a homozygous, the variant, the percentage of reads would be close to 100. So, for germline variant, the American College of Medical Genetics and Genomics and the Association for Molecular Pathology has come up with what is known as a ACMG classification. So, and based on this classification, you can classify variants right from the benign, likely benign, to likely pathogenic and pathogenic. So, and there are different criteria by which these variants are classified. So, for example, there are different levels. So, the first important is the population data. So, if that particular variant is present in a very high frequency in normal population, then the likelihood of its being likely pathogenic or pathogenic becomes very low. Whereas, if that variant is rare in a population, then it is more likely to be associated with and if it is also consistent with the phenotype of a disease, then it is more likely to be associated with that disease. Then there are computational and prediction tools. What do they predict about the effect of that variant, either on the protein function, protein stability, or if it is binding to some other protein, how is it affecting that? And there are different tools which are available to do. Some are what known as meta tools, where you combine a lot of tools into one, or they could be standalone. For example, SIFT, Polyphen, they are standalone, but there are other tools which combine scores from different prediction tools and give a score. So both of those kinds of tools are available. And then there is functional data. Functional data means that someone has already had that variant in a patient or in a or they created that in a cell line or something and looked at the function, what that variant is doing to the function of that of the protein encoded by that gene. And a functional data is always very good to have because it then tells you that this variant is actually producing a pathogenic effect or a lethal effect on the proteins, either the quantity of the protein or the function of the protein. Then segregation data, for example, if you have a family and where multiple individuals are there and the variant is segregating only in the affected and the ones which are affected are either carriers or in a context of autosomal recessive disease, then also it's a positive sign that it could be a pathogenic variant. Sorry. De novo variants are important for autosomal dominant diseases. So autosomal dominant disease, if you have a variant in a child, but that variant is not present in either of the parents, it's a de novo variant. So that variant is upgraded. But the only caveat here is that there could be variable penetrance. So in some of the autosomal dominant diseases, it's a problem that the father and the mother or the child or the father and the child may have the same variant, and the father may have only no symptoms at all or very mild symptoms, whereas in the child the symptoms might be more severe. So that can be an issue, but normally if your variant is de novo in the context of an autosomal dominant disease, then it's more likely to be pathogenic. Then allelic data is also important, and then there are other databases and other data. But the most important ones actually are the population data, the functional data, the segregation data, and the computational data. So what are the key sources of information? I think ClinVar was already referred to. So ClinVar is it's a repository. So for example, if I find a variant in one of my patients, I submit that variant. So you have to make an account and then you submit that variant to ClinVar. So ClinVar is and anything which gets submitted to ClinVar doesn't become pathogenic automatically. You have to provide evidence why you have classified that variant as benign, likely benign, VUS, likely pathogenic or pathogenic. So ClinVar is actually a public repository where people have submitted their data, and it's very useful for clinical phenotypes. So if someone has already reported a variant as pathogenic and has provided good functional evidence for that, then it's a strong point that the variant is actually pathogenic. But sometimes there is a conflict. So like there are 5 submitters, 2 say that it is pathogenic, 1 says that it is uncertain significance, then the problem occurs. But if there is a consensus, like there are 10 people who have uploaded that variant with a particular phenotype and all of them are calling it likely pathogenic and pathogenic, then you're good. Like, okay, this particular variant fits with the phenotype and it's likely to be the disease causing variant. But when there are conflicts of pathogenicity, then it becomes a problem. Few people calling it pathogenic, some calling it VUS, then it becomes problematic when there are conflicts. Then LOVD is the Leiden Open Variation Database. That is also a very good resource where for a lot of genes, the variants are uploaded on that database, and it's also free to use. So you can look at the variant that you have found on this Leiden Open Variation Database. Then there are other tools which are commercial. So HGMD, you have a basic version, but the licensed version, you get more features. So HGMD is one. Then Genomenon is another, and there are others as well. But there are some things, some data sharing initiatives like Beacon, but there is others like Decipher. So those are also you can use. And then there are some computational approaches like Intervar. So there are gene level databases and variant level databases. So what are gene level databases? So gene level database, one of the very important is OMIM. OMIM is a database where you have got both phenotypes and genes associated with those phenotypes. So OMIM is online Mendelian inheritance in man, and it's a very good resource where you can look at a gene and also the phenotype. For example, with one gene you can have different phenotypes. So in OMIM, the phenotype is before the phenotype you have a hash sign, and before a gene entry it's an asterisk or a star sign. Then ClinGen was the initiative which was started to ascribe gene association to disease phenotypes. And then you have got others like Decipher, Orphanet, etc. And as I've said, ClinVar and HGMD and LOVID are variation databases. So these are portals where you have got different variations which have been reported by submitters for different genes. And then there are case level databases where both genotype and phenotypes are shared. And again, there's something known as matchmaker exchange. For example, if you have got a variant in one particular gene, you upload it and you ask people on this way, it's like a matrimonial site or something like that. So that's why it's called matchmaker. Someone else has found that particular variant in the same clinical phenotype or a different phenotype. And then in the US, there is the NHGRI is one of the resources for case level database, and the UK Biobank is also one of the other case level database. And NORMAD is very good resource which a lot of us refer to. So this is the genome aggregation database. It was earlier EXAC, now it is genome aggregation database, and it's also got versions. I think the V3 is the current version. Is it right? The V3 is the current version? Oh, okay. So I found this very interesting. I mean, like, if we could sequence everyone, all the 7 billion people, then we would find any known variation in any particular nucleotide in all of us. So that is the power of sequencing more and more individuals. I thought it could be a good way to resolve more VUSs, I guess. So these are the three main. So this EXAC is no longer there, and the current version is the genome aggregation database 3. And actually, this is a little outdated. It's still 2021. It keeps updating. You can go to this genome aggregation database was from the Broad Institute. But you can see that in the version 3, they have tried to include more racial ethnicities. So there was a concerted effort that people from other ethnicities should be sequenced, so that normal variation in people of other ethnicities other than Caucasians could be captured, because it's very difficult. Like if we say that we found a variant here, but I mean what is the frequency of in that particular ethnic population is very important. Not that you will compare a variant which is found in a Southeast Asian or the Middle East Asian with a Caucasian or Afro-American. So there was a concerted effort. So mainly Afro-American there was a significant increase in the NOMAD V3 version, and also people from Hispanic ethnicity, and Middle East and South Asians was almost similar. But there was a concerted effort to get people from other ethnicities also to be sequenced. And in terms of India, we have got now 10,000 Indian healthy individuals from different parts of the country. The project has already been completed, but it's not available for publicly available. But if you write a project or a research in a research mode, you can query that database as well. So this is one of the genes. How do you can search on the NORM AD browser. I think I'll not go into details of that. Skip this. So the ClinGen is a very important resource, and that defines the clinical relevance of genes and variants which are for use in precision medicine and research. So the ClinGen was developed as a semi-quantitative framework to assess the strength of evidence for role of gene in disease. For example, if you find a new gene for a particular phenotype, So what are the how can you ascribe that gene to that phenotype? You should have some framework or evidence to say that this gene, this particular gene, which you have, a new gene which you have found for a particular phenotype, is causal for that phenotype. So they made an evidence-based framework for the as a clinical genome resource. For example, if you have sequenced a patient with a particular phenotype and you found a novel gene, which has not been reported before, you can't just say that this is a new gene associated with the phenotype. You have to have a evidence-based framework to show that, and this was what was done by ClinGen. So as I've said that the purpose of these two, the ClinVar and the ClinGen, is different. The ClinVar is a database for sharing variant level knowledge, so that if in a particular gene, if you have found a variant and you have classified that variant based on the evidence that you have as benign, likely benign, view as pathogenic or likely pathogenic, that is submitted in ClinVar, whereas ClinGen has a different purpose. ClinGen is for gene level information. So it's for ascribing genes to particular disease phenotypes. So this is a gene level database and this is a variant level database. Although in ClinGen also they have got now variants as well. I mean they have got disease specific experts who classify variants as well. So this was the criteria which is used for classifying, ascribing a particular gene to a phenotype. So there are different points that for classification. I think I'll not go into details of that. And ClinVar, as I said, ClinVar, how many of you have submitted variants to ClinVar? Okay, oh, good. So you routinely do that?
Yeah, I submitted a lot of them.
So you do it in like sheets, like batches, and because like because doing one will take so much time. So
We've done it by batches.
okay, and who is responsible for that? I mean, like you do it like,
Well, that was
and how frequently do you do that? Per week or like?
no, we don't do it anymore.
Why?
Because that was part of the Mendelian genomics project that we had,
Okay.
where Gene Matcher was created and other tools were created. So part of our responsibilities was to submit everything that was produced during that project.
Okay.
So it was part of that, but we are not responsible anymore. It's the investigator now.
So the investigators do that now? Like,
I'm assuming, but I don't know
okay.
how to do it. So something that I don't follow up.
Okay. So the Leiden Open Variation Database is another database, so which you can use for variant level. And this human genome gene mutation database is also very useful when you want to look at variants. So it has also got like a free version and a licensed version, and there are more features in the licensed version. For people with institutional ID, the free version is the free version you can access without any, but for the licensed version you have to pay a fee. So again, these are the annotation databases. The annotation tools I've said, you can use NOR, you can use web for annotating your VCF files. Cosmic database is for your cancer, somatic variants in cancer. ClinVar and HGMD is for germline variants, and for functional consequences, SIFT and polyphen, and then you have got this CAD scores as well. So these are some of the resources that you can use. I put the IDs, I mean the HGMD, OMIM, exact is no longer there, so it's NOM-AD. GTX is your gene expression portal, SIFT, polyphen, and then if you want to look at the same genes in mice, orthologous or the similar genes in mice, the Jackson lab has got a very good resource. So I think I'll end here. But this is part of a talk where I looked at reanalysis of exome data as well. Thank you. You want to put?
So we are talking about different databases of annotations, and we're talking how much agreement or disagreement they have between them.
There's a lot of disagreement.
Well, there's a lot of strict disagreement.
A lot of what?
We'll see. I'll show you in a second.
Okay. Yeah, yeah.
Oh, I'm not going into many details, but so we were talking about how much agreement there are between annotations. So it's not my slide. So this is a slide from actually the authors of chess, which is another type of annotation. So they were trying to show these are protein coding transcripts and how much strict overlap are between them. I don't remember how they define it. I think it was, based on the introns, the starting and ending positions of introns and all that. As you can see, there's not much overlap, right? Well, first there are more annotations in one annotation or one type of one of the annotations types, right? So some of them have more, others have less. So starting with that, you're not going to have much overlap on those. So it's quite striking, right? I don't think you're going to go wrong using one or another, though.
So
Where is this test from? I never heard about this test.
I don't even sure. I don't remember. I remember there was a striking, and I said, okay, let me store this slide because I thought it was really cool in the way they show it. Again, I don't remember either what it means, a precise match. I mean, they could be really strict, right? If one base is off, we call it's not a match, right? So a base is not going to make a difference in your analysis.
and not just that, actually there are one other issue which a lot of gene annotations have is how you define gene. So there are small peptides, there are many small peptides are there which are transcribed in genome. And there's one, I don't know how much is true, but somewhere I read that almost 92% of genome is So almost 92% of genome transcribed at some point in some cell based on the sequencing data. How much that is correct is we can debate about it. Oh, okay. So basically, almost like 92% of genome is transcribed at some point in the cell fate somewhere, some condition. That means most of the genomes are transcribed in. Now, when you define a gene, so as a molecular biologist, the first thing when you think about gene, you think like it is a hard line, like it is like boundary of Chandigarh. You know that it starts from here and here, and there is a survey stone is there which says that welcome to Chandigarh and goodbye to Chandigarh. That is not true. The more and more you actually look into the bioinformatics space, this is a very fluid area. The way they are defining genes, and it's actually a lot of assumptions are there, and based on that, they are just all the models that predict. So because a lot of these things are not either based on transcriptome data also. The genes, what they are talking about, whether this is a gene or transcript or all these things, are not often supported by or even sometimes supported by some random sequencing projects, things like that, but not always completely supported in that way. And start and end of the genes are not properly supported. So all this one, which is prediction models, for gene prediction models, they rely on certain assumptions. And one of the things that confound this is how you deal with small peptides. So they say, okay, if you have, say, 40 peptide, small peptide of a transcript that can create a 40 amino acid peptide, I will consider that as a coding gene. And then that means now you will inflate this whole data into 100,000 genes because those peptides are also now coming under the gene. But some people will say, no, I will not actually consider that anything less than 200 amino acid will not be considered as gene. So there is a huge underway assumption is there based on which the gene numbers actually are defined based on model. Now, in bioinformatics, there is nothing called as right or absolute right. Okay, so for most of the main genes, like there will be always agreement. Like what Francis was showing, around 23,000 to 30,000 genes will be agreed upon by every single database. And after that, whatever left is something that you can always debate about. The question is, are we actually do we have any kind of biological insight to make sense out of those genes which are predicted by one database versus other? In fact, The truth is that even we are not able to make sense out of 30,000 genes, then those extra, no. So that means like we are fine with using any of these databases. But the only requirement is we need to write that when you are publishing your paper or when you are actually giving a talk or anything, like you need to actually say, I have used this particular database, this particular reference or the build of this database, and based on this, my results is this. So, so your analysis is actually, analysis when you are doing, that actually needs two things. One, the database, the complete reference of the database, its build, and the complete reference of the version of the softwares and its dependencies. The moment you have all these things, because again, the moment your analysis software, it changes its version, you will see a small change in the in your results. And sometime actually, like if you are doing like a functional analysis where the Go terms database is getting curated, and if you do something 1 year after, and you will see a completely different result. So till the time you are clear about your references, what kind of reference genome you are using, which database you are using, which version of software you are using, and properly put that in your method section so that any result that you are publishing is true under those circumstances or those under those thing. And then if you change something, there will be a small changes. But again, there is another aspect of it. Any biologically significant changes you should see irrespective of your the smaller ones, that are the one which actually comes and goes in case of you are changing your software. So those are the two things. Always please be aware of what build you are using, what references you are, version of software you are using, document it, write in your method section, in your thesis, say like this is what I did, so somebody else want to reproduce your results. And Francis has shown the Conda environment, containers, Dockers. So Dockers also help you to share your codes and along with the version control, so that means somebody else can replicate your analysis. So those kind of things are actually part of a good practice with the bioinformatics. And always write code clearly, document it, file name should be explicit. Even like when we are doing R, we are doing, so when what the objects what you are saving there, give them names that you can actually recognize later on what those names are. So some of these are the good practices. Comment it out, like add as much as comment possible, because you are also sharing some of this to codes with somebody else who wants to verify that what you did is right. So that is something that you need to be aware of. Okay. So don't worry about what number of the what is the true number of the genes. Nobody knows. That is the reality. But we definitely know that we have some 23,000 to 30,000 gene in human genome, and beyond that we can debate. .30. So the thing is like tomorrow.
We said 6:30.
Yeah, if the lady that was screaming in your in your corridor, if she's still there, we can come at 6:30.
Well, if you're lucky, we can sleep until 5 today.
Yeah, so I think tomorrow we'll see differential gene expression analysis.
Will it take a lot of time? So we can start at 8 as well.
No, fine, 8:30 should.
We keep pushing it earlier. 6:30 on Friday, I'm telling you.
Yeah, I mean, so I think the most, so I think tomorrow will be okay. Tomorrow will be a,
Thursday will be the bad one.
Thursday and Friday will be really the bad ones.
Then we can start at 8 on those.
So, yeah, but thing is like, so tomorrow is differential gene expression. You have already the count table, and then now you are looking at various scenarios of differential gene expression. We'll see like if we need to move one of the Thursday's talk into tomorrow, so that if that we do, then we have a little more breathing space on Thursday also. But Thursday is something then we are going into SRAT, single cell, a lot of new concepts will be there in that. And then Friday will be like completely mayhem, because we will have a, we'll have the final battle, and whoever survives, we will, with head, we will allow them to go back to their homes on Friday evening. Yeah, but it's fun, like And I think you guys did a great job by creating count table today. So this is something
And we have a post quiz as well.
we can have an examination.
Oh yeah, you should do that.
A small quiz we can have at the end, so that we know how much they've learned.
Nobody will come on Friday then.
I don't know if I want to see the results of that.
No, they will do good. They will do good. Actually, see, they all of them created count files. Up to that they did. So, yeah. See, I was telling this to somebody.
It is easier to do on a Jupyter than to do on a RStudio.
It's just matter of convenience. I mean, there's no difference between RStudio and Jupyter. Like, it's a.
Everything is downloaded there. Like
So the good thing.
looks like more convenient to do it there.
You know, good thing about Jupyter is that you can run any kind of script within the Jupyter. Like you want to run Bash, you want to run Python, you want to run RStudio. RStudio can only handle R, and then I mean you can do other scripting, but it's more complicated to do that within that. So that is why Jupyter Notebook is a good, and they're learning that. So I think you see, yesterday it was a tough thing. You know, you saw the Jupyter Notebook and you were afraid of it. Today you are getting the feel of it. By that time Friday you will be more comfortable with that environment, that the way that you are handling it. So I think slowly you will get more and more comfortable. One last point I wanted to actually mention. So Francis is coming from the computer science computational background. There are other people who are from computational background. But for a biologist, because I was somebody who never got trained in any computer language or programming courses. But the way I
lot of us here are forced to.
no, but that is how we all.
I'm a biologist, by the way.
Yeah, yeah. But
Like we don't have a bioinformatician on board. In most of the departments who are doing clinical reporting. So sir खोल के खुद ही सीखना पड़ता है.
that is why. So, but the best way to do when you are doing this kind of complex analysis. So, Francis gave you a set of code. First 6 months, just change the file. So, put your file into that folder, change the file name, run it from beginning to end, generate data. Just get comfortable with running that, and then create your data. You do whatever you want to do. So what you are doing, you have a set of code. You are not playing anything with those codes. You are just changing the file names. You are bringing in new files, running them. After 6 months, when you are used to running them, then go and slowly modify it. Then what will happen? After 6 months, you can go and check out in say, you can ask ChatGPT that, hey, I have this bunch of codes. Can I actually make it in a loop? Then ChatGPT will modify that code, and then you can run and see like how it works. But if that you want to do today, it may not. It's hard to do. So first get familiar with this. Spend some 4, 5, 6 months on this. After that, you start modifying that code. Then start writing your own code. So it's a process. If you do that first day itself, writing the code, even if you are taught, you will not be able to do. So first learn how to execute a batch of code with your files. Make sense out of it. Then see that things are actually run rightly or not. All those things. Do it for 6 months. And you are doing your analysis. It is taking a little like primitive way of doing it, but then still you are getting your data out, you are seeing your results. Once happens that, then try to actually combine those codes, do some loops, pipelines kind of stuff. Then you saw enough core, start running some enough core pipelines and see if you can run them, because now you have a feel of running some codes. And once then you start working on more sophisticated. And at that point, you can also go and do some courses in Coursera or Udemy or something like that, which will give you more foundational idea about R or Linux or Bash scripting. So
Code Academy, like the workshop, pre-course material for the workshop.
no, I'm saying some of these things actually, so it has to be done slowly and continuously. And this is not going to happen in a day, especially for biologists and especially for people who are already in lab or who are already master students. So what happens for them is like you don't have that much time to put like hours on this. So whatever time in hand, but then keep engaging yourself with this exercise. Don't leave it. Like don't say like today if I am doing it and then I will do after 1 month, then nothing is going to happen. You will forget everything. And most of the time it is muscle memory, actually your fingers. It's not the brain that actually works. The coding, and so when you stop doing something and then after like 15 days, you will not remember what syntax is. Because it's not your remembering, so your fingers are remembering them.
ChatGPT is dangerous. Though.
Yeah, ChatGPT is dangerous.
The muscle memory is gone at that time. You just copy and paste what ChatGPT tells you.
Yeah, because actually the problem is you are defining a problem, it will give you a code and you are just running that code. So it is, after some time, your ability to even troubleshoot will go away. You are not even, nowadays a lot of people what they are doing, they will run a code, and if some error happens, they will copy that code and put it to ChatGPT and post a debug it. And ChatGPT debug. So they are not even spending a minute at looking at that if I'm missing a comma or something or not. And end of the day, you will lose that skill. And I mean, we don't need too many gray cells, but definitely we need. No, this is a real problem, and I think. But yeah, this is also a useful tool for at least beginners also, that you can. I was fortunate because I had few people who were.
It can help, but you should not blindly trust it.
No, you can trust, but the problem is that it become more like a spoon feeding. You are just, they are giving you the solution. You are not struggling to get that solution. No, but so that is the only way learn. So the way we do, when I used to learn, I write a code, the error comes, I will actually copy and paste that error in Google, and Stack Overflow will be having, some people have said, oh, you do like this. So a lot of those things don't work. So you try few different things and then something will work. So every time you are interacting with that code, you are learning something, okay? And one more thing actually, which for my personal experience, I learned, teach somebody. Whatever you learn, teach it. Teach somebody else, one person. You will learn by doing that, because a lot of things that you haven't thought about when you were doing it, you will think when you are saying to that person, like, okay, hey, and then this person will ask, oh, why you have to do like that, and then you will think, and then you will actually try to figure out. So teach somebody, so one of your colleague who is not in the workshop, and that will help you to learn all these concepts. So reinforce this, keep doing, teach somebody, and then try to troubleshoot, and then, and it's possible. Believe me, 5 days, today, I have seen you people who are more comfortable than yesterday. And then as day progress, I believe like by Friday, you will be really comfortable with Jupyter environment. At least if you are not ready to write your own code, but you will be definitely be able to execute the codes, and then you will be able to do the analysis. So that is what, but just keep touch of it. Don't leave it. Okay. 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 So see you all tomorrow. Is anything else, Francis? Okay. So let's meet tomorrow 8:30, and then we'll start from day. Tomorrow is one of the most important day for RNA-seq. Yeah, because that's where a lot of conceptual information is there. No. So Francis is going to start with bulk sequencing, but single variant analysis, multivariant analysis, and all, like how you incorporate models and how you do various things in that. So a lot of things. Basically, that is the backbone. Main stuff for bulk RNA-seq. Yeah, and up to this portion, like alignment and all this, count data generally not, but before count data, this is something you need to do for everything, irrespective of whatever technique you are doing. You do FastQC, you do the alignment, you do the trimming, all these things are done for everything. So it's something you have to do for, even if it is epigenetics, it is variant, anything that you have to do that. Okay, chalo. Anyway, have a nice rest of the day. See you tomorrow.